I put together some slides of what I think are the salient points from the day, and then also some examples of how you guys, the industry, are taking part in answering some of those questions. So when I say I put together a talk of the salient points of today, I did it at 10 o'clock this morning. So if you say, I don't remember a speaker having said that, this is my opportunity to make sure they do get to say that. So that's the way I do things. I let them talk all day, and then I spend 20 minutes telling you what I want you to remember when you walk out the door. So the utilization of genomics, or the adoption of genomics, the integration of that into our national cattle evaluation isn't a single breed application anymore, in the sense that there are multiple breed associations that have adopted this and are working very quickly towards that end. And the methodology with which breeds are doing it vary. But the point is, the outcome is still marker-assisted or genomically enhanced EPDs. And really the shift is moving away from having commercial partners to do this in terms of some of the companies that we're used to talking about, and really breed associations deciding to control that information largely on their own. Like all technology, cost is declining. I remember when I left the University of Georgia in 2006, I bought my first thumb drive, and it was less than a gig, and it cost me, I think, $45 or $50. Now you go to a meeting and they give you a gig thumb drive for free, right? So think about that in the sense of genomics. Things are getting cheaper, and I don't want the industry to be hung up on the fact that it's always going to be too expensive to deploy. By all means, use it judiciously, but don't get hung up on the fact that it's always going to be too expensive to deploy. And even, and again, a comment I made earlier, even HD assays are cheaper than some phenotypes. Methods have evolved and are still evolving, as Mark talked about earlier, and ways to be able to get genotype information much cheaper in terms of parentage for large groups, perhaps targeted low-density panels, and then, as Mark talked about as well, sparse genome scans. And I think that's extremely exciting as you think of how quickly these types of things have evolved and the applications that could be there because of it. Reduced panels, and Allison talked some about this, have been very effectively used in other species. And a lot of that is because the ability to use high-density assays to be able to reduce that in terms of panels for targeted traits, and now the development perhaps of evenly spaced LD panels that we can then impute up. So you can think of it as maybe HD quality at Walmart type prices, right? The ability to garner more information. And initially that's what we had in the beef industry, was really a low density panel in terms of Mary L. Igenides 384 that the American Angus Association first adopted. From Allison's talk again, we know that some of these panels do an extremely good job at tracking family relationships. And that's not all bad. We just have to understand the limitations that perhaps that provides as we go over time and across different populations. A low density assay, so that's what I mean by LD, about a 9K chip, could add value by allowing a lot of different uses. And something else that Allison talked about before, it's a pain as we think about the dollar value that's been spent on the US on a couple of causative mutations for genetic defects. And if we could have spent that money on genotyping, where we got a lot more bang for our buck, and I think that era is coming to us relatively quickly. As we grow the size of our discovery populations, it really enables us to make better use, perhaps, out of some of these LD panels, and again, the concept of imputation. The MBV accuracy decay, and it's been alluded to by several speakers and discussed in depth by Megan. We know that these tools are not robust across breeds, and I think that's been fairly common knowledge for some time, but I think Megan did a nice job of discussing how it's not robust across time either. What she showed you was in years, 8 to 10 years, and we begin to see some kind of decay. We've seen data in beef cattle and also in other species where maybe 4 or 5 generations, and we see some decay there. I think along with that, we need to ask ourselves, as we continue to develop these training populations over time, the really old high accuracy animals that are contained in training, How much use are they? If they become very genetically distant from our target populations, their genotype, their high accuracy, but are they still descriptive of the animals that we're trying to predict? I think we need to be aware of that and contemplate it as we go forward. Retraining is going to be needed. And again, being able to have access to the genotypes is crucial for that. So you have more flexibility to retrain and redevelop those when you actually have access to the genotypes. The high-hanging fruit, the things like fertility, like feed intake and feed efficiency, we need reliable phenotypes to be able to develop these genomic predictors. There's no way around it. And for things that are expensive to measure, and I'm going to throw this out to you, do we need to consider the concept of strategic phenotyping? As we go through and once we develop, say, a prediction set for something like feed intake and feed efficiency, that's not good from now till the end of days. We're going to need to reevaluate it. And for a trait that's expensive to measure, we really need to give some thought into what animals we go in and collect phenotypes on. If, for instance, I have a grow-safe system and I say, you know what, I'm a limousine breeder and I'm going to have my bulls there, and let's say Dave, for a day you're a limousine breeder too, and I'm going to invite my buddy Dave to feed bulls there, and that's the set which we're going to evaluate genomic predictors and develop genomic predictors. Well, Dave and I are really lucky because it's going to work well on our animals, but the rest of the breed may not be so lucky. So we need to think about how we develop those phenotypic resource populations. As we increase the number of traits, economic index selection is going to become even more important. And selection on profit, folks, which of course is revenue minus cost. I'm going to throw this out here, and people are either going to be mad at me or they're going to nod their heads in agreement, but secretly they're going to disagree with me. Select the ones that make you money and learn to like the way they look, right? We need to perhaps adopt that in beef cattle and think about selection on true profit. Data recording is obviously critical. If your breed's national cattle evaluation is behind the game in terms of data recording, genomics isn't going to help catch it up. More and more SNPs. Steve Catchman talked about this. Genomic predictors in some cases are better based off the HD, but not substantially. There's a diminishing rate of return. We can look at using a 128 versus a 50K, see a tremendous benefit there. But then 50K to 770, perhaps not quite as well. But maybe we haven't been using the information off the 770 in the manner where we can actually garner more from it. And I think that's an important thing to think about. I showed you that square peg in the round hole. Maybe that's what we've tried to do with the 770, is force it into the paradigm in which we've been used to using genomic information. And so I'm not saying that the 770 is going to be applicable, perhaps at the industry level, but perhaps there's tools that can be garnered from it that will be. And so I want to leave you guys with that as you think about it. Sequence data, defects. And Tara showed you being able to identify what could be construed as a defect for infertility off of the 770, or using 770. Sequence data will probably allow us more as we think about loss of function mutations. And so a question was, how much would a test cost for one? I'm not worried about the one. I'm worried about what we do. I'm not worried about finding them. I want to find them. But I'm worried about what we do in breeding cattle systems when all of a sudden I give you 300 or 400 that impact fertility. How do you guys use that? I think that's the important thing to contemplate. And we need optimization programs that contemplate those kinds of defects whilst also trying to optimize or maximize our breeding objective. So we need to become a bit more sophisticated in terms of our breeding strategies. Mutations, again, kind of Tara's talk, infertility, and that's nice. Fertility is a real problem for us to try to select for and improve. And man, if we can find the mutations perhaps that have a large impact on infertility, that can give us a lot. And what it really does is we think about fertility in terms of overall production efficiency. That's the trait we really want to improve, is fertility. So here's something that I think is interesting from Beef Magazine here, and you guys have probably seen this before. Do you feel like you have a good understanding of genomic or DNA information being offered by seed stock suppliers? right? And this was in 2010, 46 or 47% of you and your fellow cattle breeders said yes, which tells me 46 or 47% of your fellow cattle breeders were really damn confused, okay? Because I don't think that's accurate. But what we need to do is get this piece of the pie larger and be confident that that's really what the answer is, that we really are that confident in using it, because going forward, this is going to be a large source of information entering our national cattle evaluations. A slide that Allison Van Eam gave to me, or I stole from her, one of the two, it doesn't matter, I have it. The problem in beef cattle, again, a nice problem to have in the sense that we're spread all over. You know, all shapes and sizes contribute to beef production in the U.S. But in terms of technology adoption, when you have roughly a third of the cows in the U.S. owned by people that have less than 50 head, right? What's their interest in adopting new technology? It's not their primary source of income. So applying these new things in the nucleus level and then allowing the benefits to trickle down and hopefully then getting some pull-through from the commercial sector saying we do want increased information is where we can get to. But obviously economics plays a pretty substantial role here. So how is the industry helping to tackle a lot of these issues? I'll mention a couple different suites of projects. One, the weight trait project, which we're meeting with those collaborators, and a lot of you are here in the room tomorrow, an industry-driven, really integrated project evaluating different genomic predictors and methodology to include this in the national cattle evaluation. Commercial ranch projects, I think of Allison, your population there in California, the population in western Nebraska, where we're able to evaluate some things like the efficacy of the HD panel. We're able to use that to identify some of the mutations and validate them that Tara talked about. So a lot of these things, including proof of concept, and then ultimately working with a group of people that are willing to try new technology and deploy it once it's been proven, hopefully will aid the beef industry. So here's a picture of those involved in what I mentioned as the weight trait project, and all 24 seed stock producers, and again, many of those people in this room who actually have done tremendous things. When I did my PhD at Georgia, I worked a lot in terms of genomics, and what did I use, Bob? I used simulated data, right? Because where were you going to get real data? But now we have resource populations driven by the industry that we can go back and look at, whether it be the commercial ranch population in California and western Nebraska, the weight trait project population, the carcass merit project, those things that we can go back and evaluate these tools and industry data before they're deployed. Because I've got a feeling that an industry wants to know that they're not just buying this free cat right here, right? They want to know that something works and it's worth it. And I think those industry partnerships and those integrated type projects help prove that, so we ensure that this isn't what they're getting. So with that, I want to thank you guys for your attention today. Been extremely good and patient audience. And I want to tell you that these talks are recorded. So if you think there's something that I didn't quite get, they're going to be online, and they're going to be on beefefficiency.org right here, the project website that Bob talked about. And I also want to point you to these other two, our UNL beef production website and the National Beef Cattle Evaluation Consortium website as well. So with that, I'd like to thank you guys once again. I hope you found it beneficial.